Hi, my name is Sarah Whiteley. I'm a PhD student at the University of Canberra's Institute for Applied Ecology and the Australian National Wildlife Collection at the CSIRO, and I study the embryonic development of dragons. The purpose of my study was to understand the genetic underpinnings of sex reversal in the central bearded dragon, Pygona vidiceps. So we know that the species undergoes sex reversal, which is when a genetic male will turn into a female during embryonic development under the influence of high incubation temperatures. But what we didn't know was exactly how this process is happening at the genetic level during embryonic development. So in this study, I incubated eggs from the dragons at different temperatures and sampled the embryos at different developmental stages to sequence the developing gonads in order to understand the changes in gene expression that are associated with sex reversal. So what I found was that even though in the end both sex reversed females and normal females will end up as functioning females with differentiated ovaries, the ways in which that happens at the genetic level is very different between the two cues, whether that is a genetic cue or a temperature-driven cue. And we were particularly interested in the genes that were associated with sex reversal because it's such a novel process in reptiles and it's something that we didn't know anything about before. And I was able to show that there was a suite of genes that are actually conserved with other reptiles that have something called temperature-dependent sex determination, in which sex is determined by temperature with no chromosome influence at all. And these genes have really interesting implications for the evolution of thermosensitive sex determination systems in reptiles. With this particular study, I was doing bioinformatics for the first time. So bioinformatics is basically like a specialized form of statistics that's used specifically to analyze gene expression data or genetic data more generally. So it was a little bit more of a laborious process because I was learning how to do this. It's quite complicated analysis and it takes a little bit of time to sift through your data, through the results, and start to piece the puzzle together. But there were definitely moments where I would get my output of genes that were expressed in the sex reversing females, and I could look through the list and I'm very familiar with a lot of the really important ones, and I could see, oh great, look, they are differentially expressed in sex reversal just like we hoped they would be, and there they are. So it was really exciting to see that. But then I could get into a lot more detail as well, and we discovered some novel candidates that we didn't know about previously. So yeah, it was really exciting. This research is really critical because it provides a foundation of knowledge that we need in order to better understand the sex reversal system in the dragons. And we're planning on doing some functional experimentation where we try to manipulate the workings of the genes that we identified in this study in order to understand what, affecting the mechanism of that gene will do to sex reversal, with the goal ultimately to be to inhibit sex reversal under sex reversal inducing temperatures, and then also to induce sex reversal in the embryos in the absence of the sex reversing cue by manipulating the pathways that we've identified in this study. So what we also found was that we had developed a model for how temperature-dependent sex determination or environmentally sensitive sex determination systems work more broadly in a paper we published last year, and this we call the CARE hypothesis. And so that stands for calcium and redox regulation. And these are really ubiquitous mechanisms in the cells of all organisms. And we hypothesized that they were the biochemical mediator between the environment and the changes that must occur in the cell in order for something like sex reversal to occur. And we see in the study that I conducted, analyzing gene expression during sex reversal, that the genes that we would expect to see on the basis of this CARE hypothesis were expressed. So that provides really good evidence that we're on the right track with this new model, and we plan to conduct experiments manipulating calcium and redox pathways in the future.